Right, our final speaker of the session is Varun Aggarwala from the Geo Institute, talking about hunt for causality, pan-camera saturation, and delineation of strains. Thank you.
Thank you. So let me start with, so what? This is the question that is always asked by our Chancellor and eminent scientist, Dr. Mashelkar, and I think this is extremely relevant for this big data meeting. We sometimes do a lot of big data, but sometimes we forget to ask, so what? And we all know, right? Like, I mean, and even I am guilty of it. Sometimes you can simply torture the data to confess anything, put something on it, and there is a huge difference between correlation and causality. That said, let me just take 30 seconds to introduce my lab and my new institute that most of you wouldn't have heard of, and then we can go to the science. So about me, I am a poster child of national education policy, started with computer science, didn't know what to do, did more computer science and statistics, moved over to School of Medicine, then went into more therapeutics at Mount Sinai, and now the focus of my lab at Geo Institute, along with a lot of collaborators and thanks to our funding agencies, is how can we alter the microbes for health and disease, and the goal is to come up with actionable diagnostics, preventive health, and therapeutics. The lab is doing pretty well. We are raising funds and we are actively hiring. So if you have interesting ideas, do contact us. The thing I want to tell is almost all the projects in the lab are totally focused towards how can we use this to impact personalized and preventive health for humans, animals, and wildlife. And we have a lot of interesting directions. With this, let me go into the talk. I will briefly discuss about microbiome. I don't think so that a lot of people actively think about microbiome, its relevance, the hype and all. Why do we need strain level resolution? Approaches for that. Why do we need to satisfy Koch's postulates? Something by which we all learn biology 101. Culturing based approaches that I worked on for strain tracking. And now something pan-genome, pan-camera saturation of gut commensals. And finally, how we can use it to delineate a lot of complex FMT trials, autologous FMT, and probiotics. That said, what is human microbiome? I am sure a lot of you know about it, but just a quick introduction. It's a collection of microbes, predominantly bacteria, that interacts with the host through immune system, production of metabolites. People are super excited because this thing has been implicated across everything. Everything that you can think of, brain health, gut health, immune health, metabolic health, reproductive health, everything. That said, this is why it's important to understand the correlation from causation. That said, there are some success stories and we will discuss. The reason why we are all so excited about it, because if this organ has a role in different things, it is very easy to perturb it. Our parents have been asking us to perturb it through diet. We can do it through probiotics or something called stool transplants. In case if you haven't heard about it, stool transplant literally means how does it read? Like you are literally taking the stool, along with it the microbes, and giving it to a person with a specific condition. There are some success stories. I will very briefly go through it and then come to the real science. So it is now the standard frontline therapy for Clostridial difficile infection. I show this slide everywhere because a lot of us working in the background do not realize how our work impacts the lives of our patients directly. So this is the endoscopy done on a patient. Tremendous Clostridial difficile infection. You can see like the pseudomembranous colitis, everything, antibiotics, nothing is working. You gave it FMT and blonde. 1 week later, everything is better. So yes, it is now the frontline therapy and there are several approved drugs. It is used tremendously for ulcerative colitis. So you can see, right, the inflammation in the colon. We will discuss the science about it and the computational methods. You can see, right, how does it look on the left, and after FMT, how it gets better. There are multiple landmark trials for it. And now a lot of physicians, even though it's not clinically approved, are using it in conjunction with biologics for refractory UC. Similarly, microbes have a huge role in women health. So huge role in bacterial vaginosis. There are super successful trials in which you are trying to alter the vaginal flora, in this case to help with vaginosis and issues of recurrent preterm birth. So it is also used tremendously. I will briefly discuss some of these results too in eradication of AMR. We are all thinking about AMR. So this is a very successful trial in which people who have received kidney transplant and who have multi-drug resistant organisms, people are using FMT to get rid of the bacteria carrying the AMR. And there is a lot of interesting thought about, can you reduce the AMR reservoirs through engineering? Not just humans, it is used extensively in wildlife too. So koala bears is a classic example. They are not born with microbes. They eat the first poop, or that's what they call pap, to get the microbes to digest the vegetation. And yes, people are doing stool transplant in koalas, so to broaden their food range, like wilding. So people, koalas from Eastern Australia who are eating particular type of eucalyptus, managum, you have to rehabilitate them in Western Australia to eat another type of eucalyptus. Bam, what do? You give them the stool transplant of koalas who are able to digest this, and through oral capsules. So again, it is being used extensively, and not just for koalas, also for dolphins. This is an extremely interesting landmark paper by some of the top people in Office of Naval Research. Dolphins, chronic gastrointestinal diseases. So yes, that's what they did. They took the donor and they gave it. So again, we will discuss the questions behind it. So again, let us get back to the science. Okay, so let us start with a thought experiment. Let us say you are doing a stool transplant for certain conditions, or even if you are consuming probiotics for that. Anything that you are going to using it to change the microbial community. So often what we see is that it is successful in some, but it is not successful in the others. And then we see sometimes the person in which it was successful, there is a consistent response, but in another person, there is a failure after that. So how to address these questions? Or at the same time, this is all happening, the stool, the FDA wants to know what exactly is happening, how is it happening and all. Or if we have to address the questions, should you do it orally, should you do it colonoscopically through enema and all, the key to answering this question lies at getting to the strain level resolution. What is exactly transmitting? What is exactly changing? Because strains are the functional unit of the microbiota. So right now when people talk about microbiome, they talk about very broad level sequencing and all. You did some metagenomics, but it's the strain, it's the actionable unit that matters. What exactly went here? How long did it go there? Did it change something? The same thing, right? If you are able to get the same material orally, then why would you like to torture yourself through colonoscopic infusions and all? And moreover, these are the keys for satisfying the Cox postulates. In case if we have forgotten about the Cox postulates, or for a lot of computational biologists, it simply means that earlier people had all these concepts that, oh, this is causing disease, that is causing disease. So Cox came up with these postulates that the organism or the pathogen should not be present in the healthy organism, should be present in the diseased organism. You can culture it out, and then if you reintroduce it, then you again get the disease and you can again culture it out. So this has to be extended to the microbiome field too. We are talking all about this is happening in microbiome, this is there and all, but the ultimate goal to come to causality, the ultimate thing that can be used for dosage to help the patients or to even come up with diagnostics rests on the concept of getting to the strain level resolution and to satisfy these Cox postulates for the microbiome version. Okay. However, strain level resolution is difficult. You may be hearing about that, oh, there are so many methods for tracking strains and all, but let me puncture the balloon right away. Those are all just big data. None of them work on real actual clinical data. And I will discuss why. So one approach for tracking strains is you literally culture the strain every time, right? You are culturing the strain from a healthy donor, from the person before FMT, after FMT, and seeing what went in, how did it change and all. However, most of the PIs here are evil, but they do not want to torture their students by making them culture everything, right? It is going to take forever. Another approach is people are doing a lot of metagenomics assembly. So you take the short reads, you try to assemble, and then come up with a word called MAG. We will discuss this as we go on. So, and then, however, this is a very, very difficult problem because unlike human genome and all, our gut or like the stool sample has reads belonging to different type of organisms. So whenever you are binning, developing contigs and all, it's very difficult to get to the low resolution bacteria. So most of these approaches that talk about strain level resolution, none of them are able to delineate multiple strains, or they are able to delineate very, very low abundance organisms. So this is the gap in the knowledge. I will briefly discuss, so this is low sensitivity, and then again, even if you do this, there are no candidates to follow up. So I will discuss very quickly the research that I did earlier and how we are trying to extend it. So the idea that we had during my postdoc was, let's try to culture something deeply once, and then we will come up with computational approaches to track it across recipients. So for example, you have a new probiotic, or you have 1 or 5 set of donors who are giving, super donors who are giving the material to everyone. Let us characterize them very well and then see what goes into different people and all. So we do this, we did a lot of culturing, and we got like in each well each unique bacterial strain, and then I will discuss the computational approach I developed on how to track it across people and why this is a difficult problem. Okay, so first of all, culturing does work. However, it's not comprehensive even to this date, and here again we have to somehow hype up our findings. So we are showing that culturing is able to capture 75% of bacterial reads, but this is important to know, we are capturing the bacterial reads. We are not capturing all the possibility of the strains. And then again, we had a lot of mouse data, so we took the human stool, put it in mouse, and then saw if from the mouse poop, because again a lot of bacterial reads may be just dead bacteria and all, we were able to find it even in the mouse poop. However, again, these are bacterial reads, not the all possible of strains. That said, we were capturing quite a decent bit. Now, why is this problem even difficult? Let's say you have a strain sequence. Now I want to find whether this is present in my gut or some other recipient's gut. Why is this problem so difficult? So the strains, they share a lot of genomic sequences with each other. So this is just, in this figure, I am just taking the pairwise distance between the strains of Bacteroides ovatus. So of course, there are multiple isolates, so some of the isolates will be 100% similar, some will be very different, but there is still a quite of similarity, right? So let's say there is a strain of Bacteroides ovatus that we want to track, it's a commensal. So it may, some k-mers of it may also be present in some other people by chance. So how to get to the most informative k-mers? So this is the problem. The problem can be reduced to, if you have a strain, can you find the most informative k-mers? And this is what we did. So we used a version of expectation maximization approach to get to it. For people who are not aware with the EM approach, the idea is you have a lot of points. You do not know the parameters of the distributions they are coming from. So you come up with the concept of a latent variable, and you use a EM type approach. That is, you start with a guess, you see whether to maximize the probability distribution, whether these points can explain the distribution, then back and forth. And this is the approach we used. Let me explain it through an example. So in this case, what we have is, we have the sequencing reads from the sample. The unknown information is, for each strain, what are the unique informative k-mers that belong to it. Once we know those unique k-mers, a strain can have a lot of k-mers, right? That's what we discussed. A sequence of size n can theoretically have n minus, in this case the k-mer size was 31, n minus 31 unique k-mers. So how to identify those unique informative k-mers which we can then track? So this is the unknown part, and the latent variable in this case was like the sequencing read assigned to each strain. So initially we come up with the best guess for the unknown parameters, and here we used a clever approach that k-mers that are already being shared with other ones, that are promiscuous, they are not informative. A k-mer that is present in my gut, someone other's gut, they may not be the unique feature for a particular strain. So we got rid of those k-mers, we reduced the set based upon this, and this is what happens. For some of the strains, we can almost clean up 80, 90, 95% of the k-mers. Some strains we cannot. And then we tried to do the EM approach. We maximize the latent variable, that is the sequencing reads assigned to each strain. by this distribution, and we used another clever approach, that is, if a sequencing read has nice k-mers and some spurious k-mers, non-informative k-mers, then likely that whole region is uninformative. So we can completely reduce the weights of the other k-mers too, and back and forth we did this approach, and we are children of lesser God. We cannot just make a claim, I am a strain tracking algorithm. We have to validate it on real data. So we were very lucky. We had a lot of germ-free mice. We gave them a list of Bacteroides ovata strains very similar to each other, and our approach was able to tell it apart, that whenever you are giving strain in all the different mouse, we are only detecting this strain and so on. We again validated it in human data. So we had around 260 bacterial strains isolated from different human gut. We were able to show very clearly and with very few reads that they are there. Okay, so now that you have developed the approach for strain tracking, so a question is, so what? Why do we care? We care because now we can use it to explain a lot of cool biology. A lot of cool biology being, so people are doing tremendous amount of stool transplants. As I was saying, right, everything in my lab is geared towards a specific therapeutics. So you have a lot of cases like this. You have some donor giving the material to a lot of recipients, or you have one donor giving the material to one recipient and so on. And this is the advantage of like big data, where if you have matched clinical records with metadata, so for each of these patients you had the metadata, the banked metagenomic sample, the stool sample, and at different time points, 1 week, 4 weeks, 5 year, up till 5 year. And some patients who got a relapse, that is the FMT was not successful, they got a repeat FMT, we had that information too, or if they got a repeat FMT from a different donor. And with this, we could identify a lot of biology, at least for C. diff patients. A single dose of FMT results in semi-permanent alteration of the recipient's microbiota. It could explain response. And of course, if the FMT is unsuccessful, rather than the patients waiting for a long time, we could tell beforehand that the engraftment is going down. So you need to, maybe in the personalized case, just take more stool capsules or get more enema doses and all. So this is the kind of approach that we want to use it to answer the question. And here I want to tell, the real world does not care about our algorithms. The real world cares about efficacy. So FMT, even though FMT remains essentially a black box, clinicians are using it. In this case, it works in 90% odd patients. In some cases, the efficacy is 35 to 40%. That's still massive. So our goal is, can we systematically predict why the efficacy is low and use it to come up with better solutions? Okay, and then based upon this, we can identify specific strains that we have patented and are now ongoing in a clinical trial at Sinai. And then we are also extending it to other complex problems. So for ulcerative colitis, this is a much more difficult problem. I will discuss this also in the grand challenges later. So here you use a mix of donors. So this is an even more difficult problem. So this patient 1001 is getting the material from this donor, this donor, this donor, this donor. This is getting the material from these donors. So now clinicians care about efficacy. They thought, oh, if one donor is not working, let's mix all the donors together. But here the problem is even more difficult. Why is, like, are there specific strains of one donor? Because all the donors have some strains of Bacteroides ovatus. All the donors have some strains of Akkermansia. And even in this case, the recipients are not that sick to begin with. They also have resident strains. So how to tell apart which strain is going in the successful people, which strain is not going, or which donor is outcompeting the other donor? So you have to use, so we can come up with any strain tracking algorithm, but eventually this is the thing that matters. I will discuss more of these results towards the end, but this is an unpublished result. So we are very clearly able to see that the responders have overall higher engraftment from the donor material. So I do not have the figure to show that. If you just look at like the species level, you will find roughly the same. In this case, it's the donor material that is coming in and outcompeting what was pre-existing. So we want to address these questions. And then a lot of other questions, that is, some patients in this trial got colonoscopy and 40 enema. Some people just got 40 enema. The engraftment is roughly the same. So perhaps colonoscopy is way more expensive. And then there are a lot of other stuff, like do you need to do 40 doses of enema, or you can do 20 doses if the engraftment is similar. But let me quickly go ahead. And then based upon these approaches, we also want to answer other type of question, that is how many strains are possible to coexist at the same time, or what is the theoretical maximum number of strains I can put in your gut. So let me quickly go ahead. Let me come to the main thing that we are doing now in the lab. So a lot of these results were dependent upon culturing, right? However, culturing cannot be done on historical data, and it is expensive, and you cannot culture every sample. So, and then if you have more donors, it's very difficult to tell them apart. So here we had this idea, have we saturated the space of commensal K-mers? So the idea is, there is Bacteroides ovatus strain, Bacteroides ovatus strain. They may all have individual K-mers, but maybe we are not going to see any new K-mer, because we have already cultured so many strains. So I may not know the exact K-mer distribution of my strain, but I know the universe from which it is coming from. So this is the idea that we are working in the lab. And here I want to say, so we want to see whether this is even possible. So how do we go about doing it? We try to look at the strains that are deposited in the public databases. But most of the public databases have a lot of wrong information. We will discuss the pairwise distances. You can see, right? This strain, this is very similar to each other, even though it is mentioned as Faecalibacterium pronidzi, but it's very different from the other strains. And if we cluster them together, we can see, right, like this strain, for some strains, it is totally different from there. So clearly, there are a lot of mismatches in the public databases. So we came up with clustering approaches to identify that, found errors in the GTDB databases and all. And then we have now these saturated K-mers. Let me show you some example of another concept. Once you have this K-mer distribution of different commensal species, the kind of questions you can ask the questions about saturation of K-mers. So here we find that for most of the species, you find 95% of all possible K-mers by just looking at 10 different strains. So yes, theoretically we have even hundreds of strains, but only 10 strains are sufficient to get to the 95% of it. And can we use that information to track? That is more than sufficient for us to track these K-mers across samples. And then we find that it is very different, like the size, the total number of K-mers. So we call this as openness and the closeness of the pangenomes. This has direct implications to how many strains can coexist at the same time. So if you are thinking of giving more probiotic doses. So these are some of the results we have, but let me come into some of the workflow. So we develop these pangenomes, we develop these pan K-mers, and then what we do is we remove the K-mers that are just shared between these clouds with each other. So we know that those K-mers are not so informative, and now we have a set of specific K-mers that we can use to track each individual donor, or if you had a probiotic, what happened and so on. So let me just show quickly some results. So earlier we were doing culturing, but let me just show here. So this is a classic example of FMT, where you did the FMT. At this point, the person was good, and then they had a relapse and you changed the donor. But I can identify it very clearly with my approach itself here, that the donor contribution is high and it suddenly goes away. Or this is the case in which you had the FMT. Initially, it was unsuccessful. Nothing went in. I also see almost very little went in. And then, bam, you gave a repeat FMT, and then it got better. So we don't need to depend upon culturing or strain tracking approaches to identify this. We can also ask these questions in terms of people are trying to address AMR by giving autologous FMT or probiotics. Are they working? How much of the microbiome they are changing? So we can compare the baseline microbes, what happens to it after taking the AMR, after taking the 7-day course of antibiotics. So this is the case where you gave them antibiotics and then you gave them their own stool FMT and it came back. And then a lot of results for probiotics, do they engraft along with the recipient or there is variation? And then we come to these complex cases too. We are able to tell it apart and yeah, so with this, let me quickly wrap up. I want to thank my lab, my mentors, and also a lot of funding agencies for supporting us in this journey. And yes, if you are looking for a position, we have some decent funds, but the goal is how can we use it to impact health. Thank you.
Questions?
Yeah.
Yes, sir.
So I was just wondering, you showed a picture of mass spec.
Yes.
And cell typing is one of the modalities that is used for identifying strains through mass spec. So have you not tried it out, or it doesn't work?
So very good question. I didn't go into why we were using mass spec. So we are culturing, right? So we cultured the stool to identify what strains can be there. But there are so many colonies. So we use all these Bruker type instruments to quickly tell what species they may belong to. So we cannot sequence every colony, right? We cannot do the de novo sequencing of every colony. That would be so expensive. We want to cast a wide net. So we did this Bruker mass spec thing to identify which species it belonged to, and for each species we chose 3 different colonies. It cannot give me the strain. It can tell me that this is likely Ovatus, this is likely Bifidobacterium breve, and then we cultured each of those wells to come up with the strains.
I see. I have another question.
Yes.
So some of these things can also be, put on a slide, and AI algorithms can also be probably be used, akin to cytology and other kind of, techniques that is commonly used in medicine, right? So did you also not try to use them?
So you mean like using AI to detect strains?
Yes.
So, but that's the same thing, right? Like there are lots of approaches that talk about, so can the reads map to some specific markers to some species and then look at the SNPs and track them? But those cannot tell apart strains that are closely related to each other.
I'm not sure, but I'm assuming that these strains would have some morphological differences which you'll be able to mark and train.
I think that might be the case, but we haven't studied them in that detail. And by definition, that entails a lot of single cultures and then studying them and then coming up with the unique markers for that. But that is like that's like a roughly important, I mean, it's a very huge expedition.
Okay, thank you.
Thank you.
Hello. Yes, good evening, sir. Sir.
Yes.
Good afternoon, sir. Sir, I am a postgraduate student from Indian Veterinary Research Institute, and lovely presentation. I really loved your presentation. So I wanted to ask you that you have done this technique for bacteria, but for the highly acidic environments, like in the gut microbes of ruminants. So can this technique, like the Kamer-based techniques, be applied to archaeas as well?
So, very good question. I have not personally extended it till there, but the proportion of archaea is still so small. So eventually, right now we are just setting it up, right? We want to see which major species were transmitted, did it change and all. Theoretically, yes, we can extend it to viruses, we can extend it to fungi, but again, it depends upon whether we can saturate their, because the working hypothesis of our approach is, I may not know the exact sequence, but I know the overall Kamer composition will come from some universe. And that universe has been 95 to 98% of it has been saturated.
Okay.
So if that is, so because we were not able to do this approach, for example, some ruminococcuses, because we were not even able to saturate it. Every time we are culturing a new ruminococcus, I think Ruminococcus nave, we are just finding that the pan genome keeps on increasing. So I had this figure in which the pan genomes were saturating, right? The pan genomes were saturating. So this was not the case. Like it was just kept on increasing. Like every time the percentage of material added. Yeah.
Okay. Thank you, sir.
Thank you.
Yeah.
Yes.
Was of course the dynamic range. That is, some bacteria are present in very few copies, some bacteria are present in millions of copies. So that's an issue in the field. I wonder, rather
So your point is, if we have a mixture, if we create a synthetic community of 1,000 strains, can my approach detect it?
Yes. Can your approach detect it equally well at the low end and of course high end you can detect? The question is, the problem is sometimes
Yes.
you mentioned about that 10% of the bacteria, 100 strains constitute about 90% of total actionable N.
Yes.
But the problem is some of the actionable N sometimes are present in extremely low copy number. So in order to detect those, you have to over sequence.
Yes. Yes, you have to over sequence, and again, if we have to bring down the cost for personalized detection and all, what we are proposing is 2.5 million reads for 5,000. We want to keep every metagenomics below 5,000, and we did these calculations. It has to be 2.5 million reads per sample. So in that case, but your question is, can you benchmark it to that synthetic community of data through simulation? I have not tried it. My hands and plates are full, but that's a very, very nice question. I mean, I don't see why it should not work, because we already know the k-mers that belong to each specific strains, right? Now we are simply seeing whether those k-mers are present in some reads in a proportion, right? I see no sense why it will hallucinate, but that is something, if I have more money and a student who is willing to do it.
Other questions?
Sir,
Yes.
sir, the detail which piqued my interest was about the vaginal microbiome. So, when it comes to the fertility, infertility, there are many reasons, like inherent reasons. Then we have the externals and like the environment, and then there is the pH difference and all. So, I mean, how are we computing the sample space of the whole vaginal microbiome?
So very good question. We are going to initiate a trial on vaginal fluid transplant, but there is a lot of things that are already known. For example, the vaginal flora is extremely simple, is extremely low, very, very low complexity. Okay. We know the healthy vagina, it's called the vaginal states, like different, I'm forgetting the name, exactly states. So the one type is Lactobacillus crispatus, one type is Lactobacillus iners. So the question is, so this is where a lot of vaginal fluid transplant is happening. So the question is, in some women, it happens and then there is a relapse. So we want to ask the same question there. Did the donor material go in? How long did it go in? And why was there a relapse?
So, but again, like, I mean, I'm specifically I'm working on PCOS. Now, again, when we look at the literature which is correlating microbiome with PCOS, we see like, okay, some strains like Prevetola species are more abundant in like PCOS women. Now, again, this also, we don't know whether because internally this is all this hormonal changes, right, mostly. And then how it is impacting the microbiome. And what is, and also the, I mean, also there is, for women, I mean, we have other factors, sexual activity, we have this pH, we have the hygiene, we have UTIs. So this creates a big sample space.
Yes, this is a very, very good question. You are talking about the baselines for different conditions and all. That is a very, very nice question. So first of all, this is in fact a question I will ask in the grand challenge. What is a baseline? Is a baseline just about a taxa, or is it about specific k-mers, or is it about specific strains and all? That is something we have not characterized yet. We know at a broad level, this is what is changing. So, but those are the questions that, but I feel once you know what is changing, you can culture that organism and then study it. So this is why we need to get to the strain or the pseudo strain level resolution, so that you can do targeted culturing. Otherwise, the culturing will be impossibly extensive.
And the second part is like the technical thing, that is, how are we then going to measure the relative abundance? Like, for example, some women might, like, say suppose like there are, you were saying like at most we are seeing the large space, right, for FMT, like all these genomes. What is the possible number of the strains which can be there?
Got it.
So say for example, we have this limited. Now it could be different for different in different healthy persons also, right? Like, but overall there would be a different abundance. So how do we compute those?
So I do not have an idea. My working hypothesis is that I am trying to model it as a bag of k-mers. I am trying to model it as a bag of k-mers, and something that we do in population genetics, where you have the frequency of it. If you have a lot of k-mers that are just appearing once, then that may be a proxy for a lot of strain richness. Whereas if you have a lot of k-mers that are being consistently repeated, then that is a sign of lower strain reduced strain diversity. So this is the approach that we are currently proposing, but for now I do not have the exact data and the results to show to you, but this is a totally open problem that people have not addressed yet.
Thank you, sir.
So I was wondering, in the computational approaches, you have an assumption that the strains are known to you a priori.
Yes, but that is only possible if you are doing culturing. If you want to do a non-culturing free approach,
yes.
So there we are using the assumption that we may not know the exact k-mer composition of the strain of interest, but we know the universe from which it is coming from. We know the pan genome, the pan k-mer from which it is coming from.
Okay, all right.
Yes, but the culturing approach was dependent upon the fact that a priori you knew the k-mer composition or the sequence of that strain.
Okay, thanks.
Yes.
Hi, I have a question here on that side. Hi. So very interesting talk. So as you said, your approach involves having a consortium of k-mers to actually predict the clinical outcomes. So are you considering peak to straw method to actually see which
method?
Peak to straw, there's a method to identify whether a bacteria is actually growing or is it a dead bacteria?
So we have not gone into any of those things. The results that I was showing towards the end, they are very, very simplistic. They are simply saying how much of the material from the donor is coming in across these pan genomes and all, but those are all the next iterations. We have not gone into that, but that's a lovely approach, the peak to trough ratio, because we know on the origins of replications, which are the specific k-mers, can we simply track their frequency. We have not gone into that. This is, there are so many low-hanging fruits that we want to harvest it before we go towards that. We have not done that.
So the idea is basically to identify which bacteria is actually growing or not growing, like or dead material.
By looking at the origin of replications and proxying it through specific k-mers.
Yes.
Yeah.
Thank you.
All right, I'm going to give the microphone to Chirag for an announcement, but first let's thank the speaker again, and let's thank all the speakers in the session.